The SARS-CoV-2 viral load really is a predictor of COVID mortality. And so this may be a lingering signature of serious, very serious disease.
Well, hello, everyone. I'm Dr. David Perlmutter. Welcome to the Empowering Neurologist. It's true that chances of dying from COVID, especially now during Omicron, are exceedingly low. That, though, may not be the issue. I think a bigger issue, or at least something that we should be considering, is that there is a reasonable risk for what has been termed long-haul COVID. In other words, having symptoms that persist even though you may be over the original big event of the virus. How common that is, a little bit unclear. We're going to explore that today. But we've now seen research that is able to predict who is at risk of developing so-called long-haul COVID based upon at least 4 important metrics that have been discovered. We're going to be discussing this new research project with a couple of its authors from the University of Washington. Let me tell you a little bit about both Dr. James Heath and Dr. Yapeng Su. Dr. James Heath is the president of the Institute for Systems Biology, and we'll talk about what that means, in Seattle, Washington. Until early 2018, he was the Elizabeth W. Gilloon Professor of Chemistry at Caltech, and for 15 years he directed the National Cancer Institute-funded NSB Cancer Center program. He received his Bachelor of Science in 1984 from Baylor and then a PhD in chemistry in 1988 from Rice University, where he was involved in research that ultimately led to the Nobel Prize. He was a Miller Fellow at UC Berkeley from 1988 to 1991 and served on the technical staff at IBM Watson Labs from 1991 to 1993. In 1994, he joined the faculty at UCLA and then founded the California Nanosystems Institute in 2000 and served as its director until moving to Caltech. His lab works on fundamental problems at the interface of the chemical, physical, biological, and biomedical sciences with focus areas of molecular biotechnologies and oncology. And Dr. Heath has published around 400 peer-reviewed studies with an H index of 122. Dr. Yapeng Su received his PhD degree at Caltech. He co-advised Dr. Heath and Professor David Ballantyne. Yapeng's PhD research resided at the intersection of physical science, biotechnology, and systems biology with a particular focus on cancer. His research utilized systems biology approaches and various single-cell technologies to tackle one of the biggest problems in cancer, and that is drug resistance. His PhD thesis was awarded with the highest honor of the Division of Chemistry and Chemical Engineering, again at Caltech. This is going to be an interesting time that we spend together learning about the approach that these researchers have taken, what it means in terms of its implications for dealing with individuals or yourself as it relates to COVID infection. Gentlemen, welcome to the program.
Thank you.
Dr. Heath, I'd like to start with you and start in a place of asking, why is this important? Why did you do this study? We're going to deconstruct the how it actually transpired, what went into it, and learn about multiomics. But why did you feel this was an important study to do?
Well, sort of by coincidence, right before COVID hit, we were in the process of designing a study on post-acute Lyme, which post-acute Lyme disease bears a lot of things in common with long COVID. When we designed the study for COVID, this was back in March of 2020, we weren't actually thinking about long COVID at the time. I don't think anyone was. But we did know, and it's the nature of how we do science, that we wanted to take patients and follow them longitudinally through recovery. And so basically, maybe a year and a half ago, we published a Cell paper on the acute disease with a very comprehensive view of it. And it turned out many of our patients kept on being patients with chronic conditions. And so we went back. We did a lot of work on trying to understand what symptoms they were having, how that connected to their electronic health records, etc., and tried to put together a deep story on long COVID.
So, Dr. Heath, your toe into the water began with the notion that people who experience Lyme disease, guess what? They don't take a round or two of antibiotics and then immediately pick up their lives, get right back into the game. That there is this thing called chronic Lyme disease that many of my colleagues you're aware of kind of deny. Many physicians in the country want to believe that, pick yourself back up, get back into the game, and there's no such thing. But what you're seeing is that indeed some people have real issues moving forward that are persistent and refractory, challenging to treat. And you were able to, by looking at a variety of inputs or variables, you were able to identify who perhaps was at risk or who indeed might have this problem.
That's right. And, one of our motivations here, because we know from patients that this is very real, and we know the frustration patients can have when they go to physicians and, it's sort of 2 aspirin, call me in the morning kind of thing.
Right.
And so one of our initial motivations was just to find metrics that we could quantitate out of the blood that these patients really had something off with them. And because, one of the challenges, as you are aware of, with any disease condition, is you need to define it. If it's just vague symptoms, it's challenging for almost anyone to think about. But if you can begin defining it through, quantitative markers, that's at least a start towards getting a handle on it. So that was really our first strategy. And then as we began to dig deeper and deeper and deeper into the data set, because we took a very dense data set on these patients, we began to resolve these, as you refer to, these factors that anticipate long COVID.
So if I may then, Dr. Su, you published a paper that looked at the differences between mild and moderate COVID-19 in terms of some of the factors that you were looking at there. And were some of those, did you ultimately become predictive? Or in other words, were you able to predict, looking at some of these markers, who might develop more aggressive disease?
So that was when the COVID first got started. Jim led this very big COVID-19 study to understand why some patients suffer from such severe disease and even have a probability to death. And some patients are just not even need hospitalization. Why? What could be the immunological differences between those patients during the acute stage of the COVID-19? And those, we find there was a lot going on related to the immune system, systematic, coordinated dysregulation when the patient go from mild to moderate and then to severe disease. And we find for our immune system, there's B cells, which are supposed to secrete antibody to protect us, to neutralize the virus. And then there's cytotoxic killer T cells, which are supposed to kill the virus infected our host cells. And there's all these other innate immune cell types that are also playing very important role in protection of us from the virus or accelerating the very bad cytokine storm or hyperinflammation, which a lot of times cause a lot of problems in our own body and contributed to the severeness of the disease. And when we look at all of those systems, different immune cells players in a system level fashion, which is where Institute for System Biology is initiating this concept when we first started. We find it's actually very illustrating. And we find actually, although many different immune cell players are playing very different role, when we look at them together, when the patient go from mild to severe, there's a very significant coordination across all these different players. And when we go from mild, non-hospitalized patient to moderate patient, which now require hospitalization, there is like a sharp change systematically across all those immune cells players. And in fact, the moderate and the severe, they are very similar to each other. So those are very important insight we gained from that study, trying to understand probably when we want to prevent from the mild to moderate stage disease progression. But by that time, we haven't thought about how those could relate it to long COVID, the persistent symptoms. But later, when we realize that's an important issue, and when we connect the early acute stage behaviors of the immune system and then the later persistent symptom, we also find a lot of interesting and important connections. So those are very important findings for us to mine the early stage of how the patient are behaving during their acute or even diagnosis stage, and how that may be related to the much later persisting long COVID
Okay,
symptoms.
well that takes us then to the Dr. Heath, the current study, and again, the risk of death is really quite low for Omicron, as it were, very, very low. And I think the death rate seems to be what people focus on. That's what the news focuses on, what people talk about. Why worry about it? Because my risk of dying from Omicron is 0.00003, as it were. But that's not the issue that you felt was important to explore, and gratefully so, because a significant percentage of people get disease, be it mild or severe, and don't necessarily pick themselves up afterwards and move on. And we in the lay population call these individuals long haulers or long haul COVID. You use the term, or the term is out there now, post-acute COVID syndrome, meaning that various types of issues, fatigue, cognitive impairment, shortness of breath, et cetera, are continuing thereafter. So you set about to try to figure out, is there anything out there in terms of the data? Is there anything lurking that our computers can try to tease apart from looking at these medical records in the cohort that you studied that would really line up with determining who will or who won't? So what did you find? Or maybe if you want to take a step back in terms of how you did it, that might be very instructive as well.
So it was a study that I think evolved in steps. So interestingly enough, initially we looked at patients when they had recovered from disease but were still having these chronic conditions. So they had what we call, almost your term, we call it PASC, and I'll probably out of habit refer to this as PASC, post-acute sequela from COVID-19. So when patients were discovering PASC, we looked at our experience in PASC. We looked in this very large data cloud that we had collected on these patients. It was actually very challenging to associate specific, we found some, but very challenging to really paint a picture of what it is in the patient's physiology that's contributing to this chronic condition. And so we started going back in time. And in many ways, inspired by some literature that had just been coming out, that at least people were thinking that maybe out of antibodies were important, maybe there were latent viruses that were playing a role. We went back and looked at the earliest time point we could. I think under the like early detection for cancer is always better. We thought maybe there's a trigger for long COVID early on, and we found, in fact, these 4 factors that not just give you a high risk of long COVID, but they actually give you risk of particular symptoms in long COVID, helping to, I think, unravel some of this heterogeneity of conditions that so many patients are experiencing.
So you then are able, based on the nuances of the 4 variables that we'll talk about in a moment, actually you're able to hone in a little bit on, in a predictive way, what types of symptoms you might expect based upon those numbers.
That's right. That's right. And I, if you had asked me ahead of time, would we been able to do that? I would have not been so optimistic. But
But
these
you would have
pack,
been a little bit optimistic, I would bet.
well, the factors that we found were so different from each other that I think ultimately you will expect that they're going to have different symptoms associated with them. Um, yeah.
So let's unpack what they are, the big four that you found. Um, Start with, let's say, type 2 diabetes. How associated was that? Were there any specific symptoms, residual symptoms that are more type 2 diabetes related? And where does it rank in terms of everything else?
Yeah, so type 2 diabetes, so we use the term how much of these long COVID symptoms or how much PASC do we explain with one of these PASC factors. And so the the three PASC factors, including type 2 diabetes, each explained about 30%. Now, some patients have multiple PASC factors, so you're going to get a number that's, when I add them all up, it'll be greater than 100%. But type 2 diabetes explained roughly a third of the of long COVID symptoms that we saw. Now, a word of caution here. One reason why we could identify that as an important comorbidity is because just in the population, there's a lot of type 2 diabetic patients. And so we had the statistics to do that. There's going to be other comorbidities, I'm sure, that are important, but one needs, we had 300 and some patients in our study. You're going to need a significantly larger study to resolve that. And in fact, type 2 diabetes, if I remember right, it seems to associate with respiratory viral-like symptoms, fatigue, things like this. Not neurological or gastrointestinal.
May I give you the opportunity to speculate as to why that may be? You deserve the option of speculating.
Well, yeah, I'm going to hold off a little judgment there.
Okay,
I think
fair
I can
enough.
speculate on the the other. We'll get to other PASC factors. I think
Okay,
I can
fair
do
enough.
some for the other PASC factors.
One factor that comes to mind, though, is seen in, this is 10.5% of the adult population in America. But one mechanism that comes to mind for me is this notion of cellular senescence being far more common in the type 2 diabetic, and really kind of across the spectrum of metabolic disorders in general, which are certainly, 88% of Americans have at least one component of the metabolic syndrome. So that being said, I think then you wanted to move into autoantibodies, and maybe you can explain what that means.
Yeah. So let me just touch back on your metabolic comment, because there is a massive disruption of patient metabolism associated with acute disease. And how that, how you tease that out with diabetes is unclear, but I think you hit it on the nose there. So autoantibodies. So those are actually the most important in terms of explaining PASC that we found. So I think people by now are familiar with the fact that you develop antibodies against the virus, and they protect you against, reinfection, at least to some extent. The Omicron is a different beast, but you still get some protection. You can also develop antibodies against anything. Autoantibodies are antibodies you develop against your own proteins. And so in principle, one could develop autoantibodies against any protein in your body. There may be, 100,000 different autoantibodies that one could develop. We looked at a very small panel, largely guided by what people knew before about acute disease. Number one, we looked at antibodies against a protein called interferon alpha 2. And so that's an important component of how your immune system basically signals in response to the viral infection. And it has a function, meaning that it can actually block that signaling. It can block the interferon alpha protein from being effective. And so we knew interferon alpha would be important, so we looked at it. In that study in which that Yapeng talked about on acute COVID, we had seen some hints of immunological signatures that were reminiscent of lupus, systemic lupus erythematosus. And it turns out the literature on acute COVID sort of began backing that up over the past year. And so we looked at a handful of autoantibodies that are associated with lupus, SLE. And in fact, and then we dug deeper. And in fact, we believe there is actually a reasonable connection between lupus and some long COVID patients. Here, the autoantibodies again looked like they associated with two things. One, they associate with sort of respiratory viral PASC symptoms, inability to exercise, things like this. They don't really associate as strongly with neurological issues. And second, the higher your autoantibodies, very striking finding, the lower your protective autoantibodies against the virus. And so they tend to not just anticipate long COVID, but maybe anticipate a higher risk for breakthrough infections.
You noted as well higher levels of Epstein-Barr virus DNA.
Right.
And we've known for quite some time that too seems to be associated, at least, with autoimmunity, with lupus, with rheumatoid, with certain forms of cancer, Burkitt's lymphoma. And in one study that came out just last month, rather than just be associated with an autoimmune condition, multiple sclerosis, the description was that perhaps it was actually causative. So how might you then tease apart this augmentation of autoimmunity as evidenced by these antibodies and the effect or the influence of Epstein-Barr being upregulated?
Right. So, as you know, chances are all of us on this call right now have EBV in some latent stage inside us. At least pretty high odds. Even if we didn't have the mononucleosis, which people as a teenager might have had, we probably have EBV that's sitting somewhere in our B cells as quiet. We know that viruses can get reactivated following things like organ transplants or certain serious autoimmune flare-ups. And so we looked for two common viruses, EBV, Epstein-Barr virus, and cytomegalovirus, CMV, both of which we know can possibly get reactivated. We only found one of those, EBV. There had been also hints in the literature by looking at antibodies against the virus that this was important for long COVID. But you actually have to see the virus activated to know that it's important. And it turns out you got to capture it right at diagnosis. It fades away very quickly, and by the time the patients actually have long COVID symptoms, it's gone. But EBV, we found, actually does correlate with neurological PASC, as in addition to other things. And so that seemed to be, and it's there in maybe in our cohort, maybe 20% of the patients. So even though most of our patients probably had EBV in latent stage, it only gets reactivated in maybe a fifth of those patients.
30 years ago, I was a member of a group called the CEBV Society, the Chronic Epstein-Barr Virus Society, when we were seeing patients who were chronically ill and had persistent elevation of their Epstein-Barr virus antibodies. And that was prior to understanding the long-term issues related to Lyme and rickettsia and ehrlichiosis, et cetera, and other infections like that. And now with this new indication that perhaps it may be playing a causative role, we also noted even back then there was some literature suggesting elevation of the Epstein-Barr virus antibodies. That's what we measured. I don't even know if the DNA technology was available at that time. Seen post-trauma, either emotional or physical trauma, that anything that tended to challenge the immune system would amp up what we were measuring then, which were the antibodies, CEBV, Epstein-Barr virus, wondering if we had DNA at that point, if we would have seen an upregulation, or perhaps a loss of its regulation, a loss of the dampening effects that normally happens perhaps by interferon alpha. I mean, who knows? But it's a stealthy little virus, this SARS-CoV-2, in that it directly seems to target, according to your findings, the very pathways that would otherwise have kept it in check. I mean, the fourth thing that you noted was the actual viral load. You're looking at levels of RNA, right? And that was the fourth predictor?
Yeah, and the viral load in the blood, as opposed to
Right.
nasal swab. That's right. That was the fourth predictor, was SARS-CoV-2 RNA in the blood. Also had an association with neurological PASC, like EBV. But also, a word of caution there. So we did talk a little bit about the fact that long COVID can afflict mildly infected COVID-19 patients as well as severe infections. Yapeng can talk about this a little bit, but the SARS-CoV-2 viral load really is a predictor of COVID mortality. And so this may be a lingering signature of serious, very serious disease, the role that serious disease plays.
Yapeng, would you like to comment on that?
Sure. So one of the factors we find is the viral load detected from the blood right at the diagnosis. And that viral level actually are not only a good predictor of patient future death, but also a very good predictor for many of the past symptoms that the patient will later suffering for longer term. And because people know that the viral load in the blood is associated with patient death, so people may be wondering, would that be only a reflection of how severe the disease is? And we knew severe disease have more likelihood to have future long COVID symptoms. So when we do the statistical analysis, we did subtract the contribution from the disease severity. And even after subtracting the severity contribution, we still find a very significant association of the viral load in the blood at right at the diagnosis, and their association with many different long COVID symptoms. And some of the immunologists who were not involved in our study, later when they were talking about it, they think it make a lot of sense. And that also suggests us for certain therapeutic insight. For example, if the viral load in the diagnosis in the blood is very related to the future long-term COVID symptom, then if we treat with antiviral drug as early as possible, if we can get rid of the virus floating in the blood as early, as quickly as possible, then we likely will reduce the chances for the patient to later suffering from long COVID symptoms.
So then I believe what you're saying is that if we could determine who has the high viral load and administer antiviral medications as soon as possible to those, if there is some triage, who have the high viral load, that we may have some effect on reducing severity and or risk for long haul?
That's one of the therapeutic hypotheses that we can potentially draw from our current study. But of course, this will definitely require more rigorous clinical testing to really prove that is the case.
Mm-hmm.
Yeah, I mean,
How
I guess,
available in the community then at the present time is the laboratory analysis to determine this viral load?
well, I can comment on that. It's basically a qPCR test, very similar to what's done in the nasal swabs. And so you would have to be a slight variation, but it can be done. You could make the same argument for EBV reactivation as well, but that's a DNA virus, which probably is telling you that you need multiple antivirals, which gets a little crazy when you start thinking about multiple drugs. It may not be good. One suggestion, and we're trying to pursue this, is right now with these two new drugs coming out from Pfizer and Merck, is we could just do observational studies, and learn if the use of these antivirals is limiting long COVID. You don't actually have to do the actively give it to the patients. So I think that's an actually very low apple on the tree.
Yeah, low hanging fruit. But let me see if I can get a point of clarification. What you're suggesting is then that in identifying these higher risk individuals for long haul issues, that beyond just an RNA COVID targeted antiviral, you're suggesting a DNA EBV targeted antiviral as a cocktail, perhaps, in that you suggest that the EBV finding being more related to the neurocognitive issues might help in that regard.
That would be my hypothesis, and I think it's pretty sound based on the data we found.
And if there is a triage on the front end to determine who gets antiviral therapy, might it not just be, at least to cover a third, just be reasonable to say, hey, type 2 diabetic, you receive?
Well, the overlap between type 2 diabetics and the reactivation of EBV or the levels of SARS-CoV-2 in the blood, I don't think we found a statistical relationship between those. Maybe Yapeng can comment, but I think we looked. And so these factors seem to be, the autoantibodies were not independent. If you have one autoantibody, your chance of having another autoantibody is elevated. But I think in terms of these viruses are EBV, SARS-CoV-2, autoantibodies are type 2. I don't think we found that there was a lot of correlation between those. They look somewhat independent.
Mm-hmm. And Dr. Su, would you like to comment on that?
Yeah, I think Jim brought a very important point. When we talk about long COVID, we just call it a single term, long COVID. But are they a single disease? Or are they just a summary of a heterogeneous different type of disease? Do patients suffer from neurological symptoms, the same type of long COVID disease as the other patient who suffer from the respiratory symptoms and other different like GI symptoms? Those are all important questions for us to think about. And the point that Jim was talking about earlier, that different past factor and the early indicators we find actually are associated with different type of symptoms that the patient later will suffer in the longer term. And that also gives us an important point to think about when we classify patients and give them different treatment to prevent long COVID. Are we preventing them from one type of long COVID symptom or another? And how those different type of symptoms are all coming into play with each other? And along that point, another important finding from our study is the uniqueness of a special symptom called GI symptom. So we find the patient that later will suffer from GI symptom when we look at their immune cell, T cells, the killer cell that are supposed to kill the virus-infected host cell, we find that there's very interesting dynamics that is going on for those patients who later will suffer from GI symptom. So generally, if we think about T cell protections, we would imagine when the patient are suffering from severe disease, they probably have a lot of host cell getting infected by the virus. So you need a lot of those activated killer CD T cell to clear the virus infected host cells. But once the disease is gone, then probably we don't need those cytotoxic killer T cells to floating around our body to kill any virus infected host cells. So generally from the textbook, we knew that the T cell after infection, after acute stage of the infection, the cytotoxic killer T cell will decrease. But for those gastrointestinal patients, they have a very interesting opposite trend. We find that there's certain T cells that are not getting activated during the acute stage of the infection. But after the acute stage of the infection, when the patient start to feeling those gastrointestinal symptoms, those T cells start to getting activated. And those are also interesting points for us to consider why certain symptom, for example, GI symptom, is related to a unique, persisting activating of immune system after the acute stage of the COVID-19.
So is it fair to say that you kind of have an opinion that the development of gastrointestinal symptoms kind of presages a longer course with this disease?
It's possible that the gastrointestinal symptom is related to this persisting activation of those killer T cells still floating around in our body. But to validate, and people has been guessing whether there could be T cells that are specific to ourselves, like the T cells that will kill our own cells. Those T cells are causing some of the autoimmune symptoms that the patient are suffering. But in our current study, we do not have the specific evidence to answer what the T cells are seeing and whether the T cells are supposed to kill. But that gives us a lot of interesting directions for future, more rigorous study to test those things out.
Understood. Dr. Heath, back to you. You indicated that being able to be predictive about who is more likely to develop long haul issues, it really underpinned why you did the study, and you developed you developed these markers that would tend to identify those individuals. You're aware that last week, Dr. Michael Edelstein in Israel was describing his work showing that being immunized, being vaccinated, seemed to be associated with a significant reduced risk of having these long-term issues. So I'm certain you have a comment on that study.
Yeah, absolutely. Well, two of our past factors we identified, three if you count this latent activation of T cells, you can directly trace back to the infection event, and so shortening that infection event, dampening it down, best way to do that is through vaccinations and boosting, is going to be highly effective. I think, if we were to make one suggestion out of this, is that, and I think it's universal, is get vaccinated. You're going to reduce your risk for long COVID. Even in a breakthrough infection, it's going to reduce that risk, I believe. It may be a little early to tell for Omicron, but vaccinate and boost. Absolutely.
Well, for all of our listeners and viewers, I'm on the exact same page as that in terms of my recommendation. Having said that, though, let's just reiterate what your conclusion is in terms of risk in the general population, these factors notwithstanding, that people should just be thinking about in terms of having long-term issues after they've so-called recovered from the initial event. How big of a population are we talking about, percentage-wise?
So there's a lot of definitions of long COVID. The most, I guess, liberal one is that if you have symptoms going on a month after your acute infection, and then there are people, I don't think this is an official CDC, I think CDC says a month afterwards, but some scientists are saying 6 months. We were looking at 2 to 3 months. One of our cohorts. The other cohort, half of them had long COVID at 6 months, long COVID symptoms. Basically, 15% of people have persistent symptoms at 6 months. That's a reasonable number, 10 to 15%.
Wow.
A much bigger number at 2 or 3 months.
That's a lot of people. And, and again, it gets back to our first conversation, beginning of our conversation, that, again, don't focus on the death rate, that this becomes an issue for perhaps the rest of an individual's life because he or she made certain choices or didn't make certain choices as it related to being preventive, wearing a mask, et cetera, and getting immunized, getting vaccinated. So let me say to you both, splendid work, contributes a whole heck of a lot to the story, and I have to say I can't wait to see what you guys come up with next because it's amazing work that you're doing. So thanks for spending your time with us today.
Thank you, David. It's a real pleasure having this conversation.
Okay, bye-bye for now.
Bye-bye.
Wow, what an incredible level of research. You talk about leading edge, and their research has led us to understand that we can be a little bit predictive now about who is at much greater risk for ending up having long-term symptoms as it relates to a SARS-CoV-2 infection, and possibly pushing a little bit more aggressive therapy on the front end. Really fascinating work that these guys are doing. I'm real interested in this moving forward in terms of what the implications might be as it relates to other areas that are challenging. As you know, they've been deeply involved in cancer as well. Thank you for joining me today. I'm Dr. David Perlmutter, and we will be back soon.